Everyone. It's so great to be at ISMB 2024 in Montreal, and I hope you're enjoying the conference. So my name is Kuan-Hao Chao. I'm a third-year computer science PhD candidate at Johns Hopkins University, and it's my pleasure today to share my genome annotation liftover project with you. And in this talk, I will introducing the new tool that I developed called LiftOn. And if you're doing any annotation liftover project, I hope by the end of the talk, I can persuade you to try LiftOn to annotate your genome. And with that, let's get started. Genome annotation is the process of identifying and marking the locations and functions of genes, as well as other important features such as promoters, enhancers, 5' 3' untranslated regions, and so on within the genome. So here's an example. It is an annotation with a single protein coding transcript composed of 3 exons and 2 CDSs. And annotation gives us the accurate boundaries of exon and introns. So genome annotation involves a combination of computational predictions, experimental verification, and manual refinement. So imagine you have assembled a new genome here, and you want to annotate it. Instead of repeating these complicated processes and annotate it from scratch, a better and more efficient approach is to lift over genes from the high-quality, well-annotated genome to your target genome. And this problem is known as annotation liftover. A formal definition is as follows. Given a reference genome R and the reference annotation RA, our goal is to transfer genes from the reference to the target T and generate the target annotation TA. And a very great example is the demonstration is the application of annotation liftover is the CHN13's annotation. It was generated by lifting over from GRCH38 to CHN13. So in April 2003, scientists announced the completion of Human Genome Project. And I quote the finish here because at that time, about 50% of the sequence are still missing or contains error. And nearly 20 years after that, the most recent reference genome, GRCH38, still contains 8% of the gaps or contains errors. So here's the diagram of GRCH38 with the purple region highlighted missing sequences due to long genomic repeats, for example, like centromere, telomere, or RNA arrays. Then the incompleteness of the human genome assembly causes the incomplete human gene annotation. And with the advance of sequencing technology, I was wondering, I haven't seen that famous photo like throughout the entire conference. A perfect ISMB is not complete without that figure. So finally, in 2022, the Telomere-to-Telomere Consortium led by Dr. Adam Philippi and Dr. Karen Megan released the complete sequence of a human genome. The T2T-CHN13 assembly added and corrected 230 megabases of sequences, which include 180 megabases of centromeric satellites and 68 megabases of additional segmental duplication, and around 10 megabases of RNA arrays. And notably, 182 megabases are exclusive to CHN13, which means that we cannot find any primary alignment to GRCH38. And after living over genes from GRCH38 to CHN13, we found 1,956 novel genes, which include 99 protein coding genes. So this effort helped us to create a more complete human genome annotation and make the annotation more complete and better. So if we're to use a CHN13 annotation, what is the tool to use? This is a screenshot from T2T Consortium Slack channel, and they recommend us to use LiftOn, which was a tool developed by our previous lab member, Dr. Elena Sume, and it was published in Bioinformatics back in 2021. And in this talk, I'm introducing a new tool, which is called LiftOn. And I will explain that LiftOn further improved the CHN13's annotation, and it further improved the annotation of mapping between more distant species. So let's take a step back. What are some methods that are available right now so you can do the annotation liftover? One approach is to pre-generate a chain file, for example, like UCSC liftover or CrossMap. However, there are some limitations because they map by homology sequence one-to-one mapping, and then it's very likely that it mapped like focusing on independent features instead of mapping everything at a gene level. So it's possible that it mapped exons separately and generate all the exons that doesn't create a meaningful transcript. And another approach is to lift over genes locus by locus. And the DNA-based approach is liftoff, and it depends on minimap too. Another approach is to use the protein-based approach, and examples include exonerate, GeneSeeker, and miniprot. Here's the summary of liftoff algorithm. So first, we extract the gene sequences from the reference genome and then map to our target genome. And then the alignment blocks, we keep the alignment blocks without the introns. And now the novelty of liftoff is that we create a directed acyclic graph with nodes represent the alignment blocks and the edges represent gaps, and a possible path represent a possible gene locus representing this gene locus. So we find the shortest weighted path through this graph to determine the best alignment combination for each gene and generate the map genes. And we can, after the initial mapping, we can do the second pass to try to find additional copies. And this way, it can find novel genes, for example, like CHN13 that I just mentioned. As for protein-based approach, the current state-of-the-art tool is miniprot. And miniprot first translates the reference protein into 6 friends amino acids and then filter out short orbs. During the initial training, it extracts 6 mers, sync mers from the protein queries and then maps those seeds in the index genome and refine the training and output the final alignment. So how can we do better? The intuition behind liftoff that we designed is that it takes the strength of both DNA to genome and protein to genome alignment, combine them and do some heuristic, and it can generate a better annotation. And we also have a thread on the X providing a 2-minute introduction of LiftOn, which you can scan the QR code and take a look. So I will go into our result. LiftOn outperformed the state-of-the-art DNA or protein-based liftover method alone that it can achieve. And also it improved the current CHN13's annotation. And most important of all, we further extended so that it can improve more distant species annotation liftover, at least as distant as mouse and rat. So before diving into the result, I want to first define the evaluation matrix. Our goal is to maximally maintain the similarity and completeness of the genes between the mapped genes and also the genes in the reference. So first, after we map the genes, we can first extract the DNA sequence and trace them into proteins. We can do sequence pairwise alignment at the DNA level and protein level. So for DNA sequence alignment, we use the standard BLAST sequence identity with the matched nucleotide over the alignment column. And for protein sequence identity, we do a slightly modification because we don't want to over-penalize the longer proteins, because it's possible that there's mutations actually inside GRCA38, or there's a genuine difference between 2 individuals. So we modify the denominator to the number of alignment column minus the number of gaps in the reference protein. So here's the result. We first generate LiftOn's annotation, CHN13 annotation, by mapping genes from GRCA38 to CHN13. And this is a gene order plot between 2 reference genomes. You can see that a gene order plot, it shows highly collinear between these 2 genomes, which is expected. And then we use the matrix that I just mentioned, compressed gap protein sequence identity. And we selected the 2 state-of-the-art tool, liftoff, which is DNA-based, and miniprot, which is protein-based. And we can see, for this scatter plot, each dot represents a protein coding transcript. So for example, if a dot is on the upper left panel, it means that miniprot produce a better annotation that preserve a longer protein and maintain a more complete protein. And then we compare LiftOn to liftoff and LiftOn to miniprot. I can see that it can improve a lot of gene locus. And finally, we plot the frequency plot of LiftOn, liftoff, and miniprot. And you can see that LiftOn produced the least truncated protein compared to liftoff DNA-based and miniprot protein-based approach. So you might have the question that all the results that I just showed you are summarizing into a single score and dots. So you may wonder how these protein coding transcripts actually look like in IGV. So I circle three dots, and these 3 dots represent the SAN transcript because it's just like located in the different scatter plot. So we can take a look at this example. In this IGV screenshot, the first track is LiftOff, the second track is mini-probe, and the third track is LiftOn. And this gene is located on the reverse strand. So you can see that the 5th and the 6th CDS, there's a splice junction here in the LiftOff annotation, but it was missed in mini-probe. Mini-probe just merged 2 CDS together. And this merge of CDS generate a lower protein sequence identity score because it causes frame shift downstream and make this protein not valid. In this case, LiftOn is able to pick the correct one from LiftOff and generate the right annotation. And there are also cases where mini-probe is doing a great job, but LiftOff disagree with mini-probe. For this example, it's also on the reverse strand and you can see that the lost CDS, the acceptor side, it disagrees with mini-probe. However, this disagreement is wrong and it causes frame shift downstream. And at this time, LiftOn is able to pick the correct one from mini-probe and generate the better gene annotation. And there are also some harder cases whether that either DNA-based approach or protein-based approach alone can do correctly. And here's an example. So here you can see mini-probe misses the first small CDS. And it is a very common mistake because mini-probe is a protein aligner, and sometimes it will think the axon is too short, so it doesn't want to create another gap. It doesn't pass the gap penalty, so it misses it. However, LiftOn generated and can maintain the valid OR. And here in this 5th and the 6th CDS, you can see that it's a bit small, so I zoomed in. You can see that the donor side disagree with mini-probe. And this difference also introduced a premature stop codon. And in this case, LiftOn is able to pick this correct one from LiftOff and this portion from mini-probe and generate the protein that maximizes the similarity to the reference protein. And the second result I show here, I want to show that we compare LiftOn's generated CHN13 annotation to the current T2T release CHN13 annotation. The blue one is the CHN13 annotation. You can see that it contains more truncated proteins compared to LiftOn, so LiftOn is able to fix a lot of them. LiftOn also has the feature to generate additional copies. So here's a circus plot showing the additional copies between chromosomes of between the target genome, which is on the left half of the circle, to the reference genome, which is on the right half of the circle. And one observation you can see is that most of the additional copies are on the same chromosomes and the corresponding 2 assemblies. And the third result, we further do a bunch of experiments to map between different assemblies. We mapped from RAFSIC human annotation to chimpanzee, and also mapped from RAFSIC mouse annotation to rat, and also Drosophila melanogaster to Drosophila erecta. So the gene order plot here, you can see from left to right, 2 genomes become more divergent and more distant. We also calculated using MASH distance and also dashing to similarity score. So here, show again the frequency plot across 3 experiments, LiftOn, LiftOff, and mini-probe. LiftOn generate the least truncated proteins across 3 experiments. So it's able to fix most of the gene locus. And also on the right is the 3D sequence identity plot. So each dot also represents a protein. And the axis is LiftOff, y-axis mini-probe, and z-axis LiftOn. So if a dot is located on the x equal y plane, it means that LiftOn preserve a longer protein and generate a better protein compared to other 2 tools and is closer to the reference protein. We also expanded our experiment to various species demonstrated in the LiftOn paper, and these species include mice, yeast, honeybees, fruit flies, veiled cress, and also rice. And you can scan the QR code to check our result in our preprint. So how do we achieve this? we introduced a new algorithm called protein maximization algorithm, which composed of 2 steps. The first step is to chain CDS together, and the second step is to search the ORF to maintain the validity of each gene locus. That's worked through the workflow of our algorithm. In this visualization, the blue transcript is the expected corrected annotation that we want to generate. And the green one is the one generated by LiftOn, and the orange one is the one generated by MiniPro. As you can see that there's a discrement in this splice junction, and also MiniPro misses this small overhanging small exon, CDS. In the first step, we extract the proteins from each annotation and do the pairwise alignment to the reference protein. So here you can see that the round splice junction introduces a premature stop codon, and it causes some misalignment and gaps in the protein alignment in the LiftOn annotation. And for MiniPro, because it misses the last small overhanging CDS, so it generates some mismatch toward the end of the protein. In the second step, we map the CDS boundaries onto the protein alignment and adjust it by the cigar string. After we have the boundaries on the protein alignment, we start grouping CDSs by accumulated amino acid in the reference protein. So here are more details. The grouping process begins with the first CDS and in each annotation and continues until reaching the endpoint where each annotation having the same accumulated amino acid. And after we reach that point, we group them together. So this is the first group. And we can repeat the process. And for example, this group is start from this endpoint and start calculating the accumulated amino acid because there are some mutations. So actually, it doesn't end up stop here. It ends up stopping grouping 2 CDS together. So that form a minimum group. So we can keep doing this until reaching toward the end of the CDS groups. And after the grouping, LiftOn calculate the partial protein sequence identity score to the reference protein for each CDS group. We can see that the third CDS group, this premature stop codon causes the 60% of sequence similarity for this 2 CDS, whereas MiniPro can preserve 100%. And in this portion, LiftOn is doing better and achieve 100%. The algorithm then iterates through each group and select the one with the higher score. And in this example, LiftOn is able to chain from LiftOn, MiniPro, and back to LiftOn and generate the gene locus that maintain a 100% similarity score. So after the initial step, even though the genes are lifted over and CDS are chained and fixed, it's possible that there are some mutations in the reference protein and may interrupt the valid ORF. So LiftOn search the ORFs in 3 frames and select the protein with the highest sequence identity score. So here you can see that French mutation, we can change it upstream stop so that it maintains the correct ORF. And there are also mutation early stop codon in the middle. So we can either change upstream stop or downstream start. And we can also fix for stop codon loss and start codon loss and so on. So in summary, LiftOn is a promising new tool for study comparative genomics. It helps you to generate a new annotation for unknown species you just assembled. And it based on DNA to genome and protein to genome alignment. Combine them and it can generate the best annotation, take the advantage out of these 2 tools. Finally, practically, we show that LiftOn improves CH13's annotation and also it can map between more distant species. Finally, I want to thank my PhD advisors, Dr. Steven Salisbury and Dr. Michaela, for their unconditional support. I also want to thank all my collaborators and all friends in Salisbury and Pertia Lab. And here are the preprint and also GitHub. I know that people read preprint, documentation more than the paper. So if you're interested, you can simply Google search LiftOn genome and the paper documentation and GitHub will pop up in the first 3 entries. With that, I'm happy to take any questions.
Great, thank you. Any questions? Hi, great talk. Did you notice like a side effect where, let's say, the inclusion of proteins in LiftOn also improves annotation of non-coding regulatory regions that may flank these coding regions?
Can you explain more about the non-coding regions annotation?
So I mean, like to the left and to the right of like the coding region, you would most likely have, let's say, some regulatory regions. I'm wondering if you looked into whether the annotations of these are also improved as like a side effect of incorporating protein alignments with liftoff.
Oh, that's a great question. I just want to clarify because miniprot, in order to incorporate miniprot into the pipeline, we need to have the protein alignment result. So for non-coding genes, unfortunately, we cannot improve that. But for a protein coding genes, for those non-translated regions, we do observe some of the stop codon loss. And actually, by searching the ORF, we can find longer proteins in those gene locus. So that's interesting. So those are the regions that we're looking into. Thank you.
Great. For the interest of time,